So I did my project on the Culex Pipiens in London underground tunnels, differentiation between surface and subterranean populations. Okay, so what are Culex Pipiens? They are a species of mosquitoes native to Europe that can cause the West Nile and Uceta viruses, along with an avian malaria and many other infections. A genetic variation of this species, known as the Mulstus form, was found in the London Underground railway system. This adaptation is due to increase in temperature underground in the winter, the ability to reproduce in confined spaces, and nutrients gathered either in larval stage or from mammals instead of birds. These traits are hereditary but are only seen in the subspecies due to reproductive isolation. So this is just like a chart that was also in the article, and it kind of just shows like where they were bred, where they like to mate, their host preference, egg production, and the life cycle that they go through. So see Mulstus and the Pipiens, and this is how it's shown. So these were the research questions that were given in the article. They are distinct populations of C. pipiens due to reduced gene flow and or reproductive isolation between the two forms. Has there been any independent colonizations of the London underground, either through trait selection or environmental changes that triggered the appearance of these characteristics? And have the Mulstus populations evolved from long-distance migrants of the surface populations from south of the testing range? So some vocab that was highlighted throughout the article is adaptation, the change of an organism makes to their environment that helps increase their chance of survival. Allele is a variant form of a gene. Homodynamy is the ability to breed continuously without a winter diapause. Homozygous is having two identical forms of a gene. Population, a group of individuals of the same species living and interbreeding within a given. Reproductive isolation, the inability of species to breed successfully with related species due to geographical, behavioral, psychological, or genetic barriers or differences. Subterranean, underneath the Earth's surface, and winter diapause, which is similar to hibernation in mammals. This is when insects become dormant in the winter and rest once it becomes too cold outside. Methods and materials. Sampling. Samples were collected from 20 locations in London where the mosquitoes breed. 7 underground populations collected by maintenance crews. Egg rafts, larvae, and pupa were collected from pools and flooded areas. 11 surface populations were collected from gardening ponds and containers of water on property closest to each site underground. In the laboratory, specimens were kept in plastic containers at room temperature, in natural light and in water, collected from the breeding sites. Each larva were given 1 guinea pig pellet as a food source, and the adults were in a container with a 12-centimeter Wattman filter paper soaked in 10% glucose, which is the energy source. When attempting to breed the two species together, the containers included a small dish for egg laying, tap water, and a guinea pig pellet. So the testing and results. If adults in the laboratory produced egg rafts, they were labeled Mulstus, and if they did not produce egg rafts, they were labeled Pipiens. After the experiment, adults were then frozen in liquid nitrogen to preserve for later analysis. Crossbreeding was attempted through placing individual virgin female pupa from the underground population in a container with 5 males from above-ground populations. A control group was also used by breeding males and females from the same population. These results were considered positive if viable egg rafts were produced and negative if viable egg rafts were not produced. The populations collected underground all produced egg rafts when tested in the laboratory, except for those at the oval location, which were then labeled as Pipiens. These collected at the Becton location were not labeled as either because no results were conclusive. The results of all genetic crosses between the molluscus populations were viable. No crosses between any males from the surface populations and underground females resulted in offspring. After 1 week of these crosses, the females were then placed with males from their own populations, and all but 2 females produced egg grafts. The offspring's allele frequencies showed that the molluscus males were the fathers of this generation. And the conclusion. Gene flow between the 2 populations is highly limited as demonstrated by observations that suggest even where there is no contact between the 2 populations, there is no viable genetic cross in offspring. This is due to the fact that underground populations are less clustered compared to those above ground, indicating a more limited gene flow in the molluscus type. The relationship between the 2 populations suggests that the C. molluscus is a subspecies of the local species C. pipiens. The genetic difference in populations are due to severe genetic drift and a caged environment, and the distinction between their forms increase with latitude. These were my citations that were used throughout the project, and that is all. Thank you.